Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series. So we all know we are having agriculture optional answer writing practice series in which we try to discuss some PYQs, some important questions from important topics from agriculture optional. So today we are going to discuss the next question, and today's question is: give an account of cytoplasmic genetic male sterility. It is written as CGMS, that is cytoplasmic genetic male sterility, and its utilization in plant breeding. Okay, and the second part of the question is also asking us about its limitations. Actually, you can divide the question in three parts. The first part about cytoplasmic genetic male sterility, briefly describe it. Second part, its utilization in plant breeding, and third part is its limitations. So this is the question actually, and this question was asked in this last year's agriculture optional paper, that is the agriculture optional paper of UPSC civil services examination in 2024. So we will see how we can approach this answer. So before going into cytoplasmic genetic male sterility, I want to give you a brief idea about what male sterility is. Male sterility. So it is the inability of inability of a plant to produce functional pollens. So this is what male sterility is. So here the plant is not able to produce functional pollens. And this male sterility is further divided into 3-4 types. So the first type is genetic male sterility, that is GMS, genetic male sterility. Then we have cytoplasmic male sterility, that is CMS. Then we have cytoplasmic genetic male sterility, which the question is asking us about. And then we have chemically induced male sterility. Chemically induced male sterility. So these are the 4 types of male sterility. In genetic male sterility, the male sterility is due to genetic regions, that is due to genes. In cytoplasmic male sterility, it is due to cytoplasmic regions. In cytoplasmic male sterility, here the genes are governed by both cytoplasmic. Here the male sterility is by both nuclear and cytoplasmic genes. Okay, nuclear genes and cytoplasmic genes. Here nuclear genes, male sterility is governed by nuclear genes. Here the male sterility is governed by cytoplasmic genes. Here male sterility is governed by both nuclear and cytoplasmic genes. And chemically induced male sterility is by the use of some kind of chemicals we induce male sterility in the plants. So these are the 4 types of male sterility. So this is a brief overview which you should know. Now let's move to the question, that is cytoplasmic genetic male sterility. So as I have just talked, it is governed by both nuclear and cytoplasmic genes. Both nuclear and cytoplasmic genes. Okay. So here we have restoration line, or you can say restorer line. So this restorer line helps in restoration of fertility. It helps to restore fertility. Okay, so these are some of the common points. These genetic. So I think 2-3 points about cytoplasmic genetic male sterility is enough. Some molecular markers is also used here in genetics. So we will not go in that much deep. That much deep. Just remember that it is governed by both cytoplasmic and nuclear genes, and here you will find a restorer line if you want to restore fertility. Okay. So this is about cytoplasmic genetic male sterility. First of all, we will see its applications, or you can say utilization in plant breeding. If the question asks utilization in plant breeding. So first of all, it is highly used in hybrid seed production. Hybrid seed production, because here male plant is sterile, so it is not able to produce pollen, so it cannot self pollinate. Here you will say that male plant is sterile, so what happens, it will not able to produce the pollens, so there are no chances of self pollination. So what it will do, it will encourage cross pollination, and we will able to do controlled cross pollination, and we can develop hybrid varieties which are good in yield, good in disease resistance, which are good in vigor. So it is highly helpful in hybrid seed production. Secondly, it increases the seed production efficiency. It increases seed production efficiency, because here we eliminate the need to emasculation. Here we eliminate emasculation. What is emasculation? In emasculation, we remove the male parts of the flower so that it doesn't self pollinate. But although here the male part is not able to produce the efficient, or you can say functional pollen, so there is no need of emasculation here. So it simplifies the breeding process. Okay, and this is highly useful in crops such as maize. Various varieties are developed using CGMS in these crops such as maize, sunflower. In rice also it is highly useful. Okay, it helps in, or you can say it facilitates crosses in self-pollinating plants. Facilitates crosses in self-pollinating plants. So what happens? It is generally used to help to do cross pollination in self pollinated plants such as wheat, so that its hybrid varieties can be developed. Okay, so it is very helpful, so that through cross pollination the adaptability of these crops are highly increased. You get the heterosis, or you can say hybrid vigor of these crops. So these are self-pollinated, so CGMS are highly helpful in getting the hybrid production of these crops. So this is one more very good use. It is highly used in genetic studies. Used in genetic studies. So it provides an excellent tool for studying plant genetics, as sterility trait can be used to trace the inheritance of mitochondrial genes and their interaction with nuclear genes. So as you see that here we have sterility traits. So it helps to study the inheritance of mitochondrial genes and their interaction with nuclear genes. So how mitochondrial genes and nuclear genes interact, how they develop, all these things can be studied here. And it also helps us to study the role of mitochondria in plant development. It also helps to study the role of mitochondria in plant development and reproduction. So it is highly used in genetic studies. Next, it helps in introgression of traits. Introgression of traits, that is, by using CMS, breeders can more easily incorporate desirable traits from wild relatives or different species. Okay, so here you can integrate traits from wild relatives, and this is very much used in very beneficial in crops such as tomato, crops such as cotton. Okay, where we get various kinds of qualities from wild relatives. These qualities could be pest resistance, drought resistance. So that will be enough. So these are the 4 5 points. I think 5 points are enough for your answer, and all the important points have been mentioned. Now in the second part of the question, they are asking us about limitations of cytoplasmic genetic male sterility. So we will also write about some 3 4 limitations. So first of all, the first limitation is cytoplasmic incompatibility. cytoplasmic incompatibility. Okay, see what happens. All CMS lines are not compatible with all male fertile lines, limiting the potential for hybrid production or hybrid development. That is, all CMS lines are not that compatible to produce good hybrid seeds. You can simply write it. So this is cytoplasmic incompatibility is one limitation. Another limitation is restorer gene. So what happens? Various times restorer gene are used to introduce or you can say restore fertility in male plant. But the interaction between CMS and restorer gene can be complex and requires careful management. So the interaction between the CGMS plant and this restorer gene can be very complex. So it acts as a limitation or challenge. And this restorer gene is used to regenerate the fertility. Okay. Many times the traits are not that stable, so it also acts as limitation stability of trait. So sometimes unstable traits get formed, and these unstable traits go from generations to generations in different plants and make those traits unstable in whole generation. So it can reduce the reliability of hybrid. So it can reduce reliability of that hybrid. So these are some of the challenges in uh, or you can say limitations in using cytoplasmic genetic male sterility. So I think 3 points I have mentioned, so that is enough. You can add points also. I have just mentioned the important points, and after that you can conclude your answer. You can conclude how male sterility or cytoplasmic genetic male sterility can act as a critical tool in plant breeding. It can help in developing high performance hybrids. It can help in enhancing adaptation. You can also mention how challenges can be fought off and stability of traits can be uh, developed in these things. So that would be good enough for conclusion, and that you should write in your own words positive statements. That is all for today. I hope you understand what CGMS is, its utilization, its limitations. So that is all for today. If you like it, please press the like button. Please do share. Please do subscribe. It takes a lot of hard work to make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.